Can you see my slide?
Yeah, I can see.
Yeah. Thank you very much. Today, I would like to present a paper named Berberine reduce pyruvate-driven hepatic glucose production by limiting the mitochondrial import of pyruvate through the mitochondrial pyruvate carrier 1. This paper was published on the 2018 in the e-biomedicine, published by the Lancet. First one, I would like to introduce what is the berberine and what is the mitochondrial pyruvate carrier 1. Oh, it's clear to see. First one, the berberine. The berberine is a natural compound isolated from this plant, Cotis cineris, and this natural compound has various type of biological activity in such as the antioxidant or anti-inflammation activity. And in this paper, they would like to check the effect of berberine in the reduce the glucose production in the liver disease in general. And the second one, I would like to introduce the MPC1. Or what is the MPC1? The mitochondrial pyruvate carrier 1. We already know that the end product of glycolysis is the pyruvate, and then the pyruvate can be converted into the lactase, convert into the alanine, or go into the mitochondria to go into the TCA cycle. So how to pyruvate can go into the mitochondria? It requires the MPC, and the MPC1 is the one is be included in the MPC complex. And the paper in the cell metabolism, such as the MPC1 is required for the MPC hepatic MPC activity, and the loss of the MPC activity will attenuate the hypoglycemia during the diet-induced obesity. So come back to the paper, I suggest they would like to check the effect of berberine in the hepatic glucose production, and they found that the berberine can reduce the pyruvate-induced glucose production through the inhibit the MPC1. So first, I would like to sorry you guys, because I choose the paper and the data, the figure is not clear to see, and I don't know why. So I'm sorry. The first result in the paper, they suggest that the berberine can reduce the glucose production in the liver. They feed the mice with the chow diet and high-fat diet, and with the mice feed with high-fat diet increase the fasting blood glucose level compared to chow diet. However, when they treat the mice with the berberine, the short name is the BBR. It decreases the fasting blood glucose level in this mice. This is a non-ester steroid ester level in the mice also be decreased. And they also check the pyruvate tolerance test and check the blood glucose level, and we can see the mice treated with the berberine also decrease the glucose level in the blood of this mice. Blood glucose level after the glucagon challenge test or glucose tolerance test also be decreased in this mice treated with the berberine. So all of these data suggest that berberine has ability to reduce the glucose production in this mice. Similarly, with the high-fat diet mice, fasted mice also increase the glucose level. However, mice the treatment with the berberine also decrease the glucose production in these mice. And more interestingly, they check the expression of some gene related to the hepatic gluconeogenesis, TGC1 alpha and G6 PEPCK, three types of gene related to the hepatic gluconeogenesis. And they found that treatment with the berberine decrease the expression of genes checked by Kipisa and Western blot. This data suggests that the berberine can reduce the glucose generation by inhibit the hepatic gluconeogenesis in mice. Next one, they check the effect of berberine in the lipid metabolism in the liver mice. We can see the high fat that induce the lipid accumulation in the liver mice. However, mice treated with berberine also reduce the lipid accumulation in mice. The hepatic triglyceride also be decreased in mice treated with berberine. This is the two type enzyme in the beta oxidation, and treatment with berberine also decrease the expression of two type enzyme in the fatty acid beta oxidation. So it suggests that the berberine also has ability to reduce the fatty acid oxidation in the liver mice. The level of the acetyl-CoA also be increased in the mice feed with high fat diet. However, berberine also can reduce the acetyl-CoA level. The lactase level also be increased, be restored in the mice treated with berberine in high fat diet mice. Similarly, with the fasted mice. All of these data suggest that the berberine has the beneficial effect in the regular the lipid metabolism in the liver mice. The continue they check whether the MPC1, as I suggested before, the mitochondrial pyruvate carrier has any influence in this condition. The first figure, they check the expression of MPC in the liver of mice feed with high fat diet. High fat diet increase the MPC expression. However, the berberine treatment also reduce the MPC1 expression. So, and the in vitro experiment, they treat the palmitic acid, fatty acid into the hepatocyte, and the fatty palmitic acid can increase the MPC1 expression in the hepatocyte. However, 2-3 type concentration of berberine can decrease the MPC1 expression. And the lactase level when the hepatocyte use the pyruvate as a major source of energy going to the mitochondria, the lactate level will be decreased. However, the 10 micromole berberine can restore the lactase level in this cell. The overexpression of MPC1 in the hepatocyte and found that when the overexpression of MPC1, the lactate level will be decreased. It means that the function of the role of berberine in the restore the lactase level be inhibited when the overexpression of MPC1. The berberine also reduce the oxygen consumption rate in the hepatocyte, such as that the berberine reduce the pyruvate oxidation in the hepatocyte. The high-fat diet mice, high-fat diet induce the expression of the phosphorylated PDH. I have to mention that when the pyruvate go into the mitochondria, it will be converted into the acetyl-CoA and go into the TCA cycle. And this step be controlled by the PDH activity. And another phase of pyruvate is go into will be converted into the osmoesterase by the PC enzyme, and then it will be used for the gluconeogenesis. So this data suggests that the berberine can maintain the activity of the PDH. However, the berberine treatment in the high-fatty mice decrease the expression of PC, such as that the berberine will inhibit the pyruvate convert into the osmoesterase and inhibit the gluconeogenesis. The acetylCoA accumulation will induce the acetylation of the mitochondrial protein, and we can see that the high-fat mice decrease the acetylation protein MPC. However, treatment with berberine restore the acetylation of MPC1. This is the data from the in vitro when they treat the pyruvate acid into the hepatocyte, and the berberine also restore the expression of acetylation MPC1. And they suggest that the acetylation and the acetylation action activity of protein in the mitochondria will be controlled by the SIRT3. So they check the expression of SIRT3 in the liver of the high-fat mice and treat with berberine, and they found that the berberine decrease the expression of SIRT3. And this is the data in the in vitro when they treat the pyruvate acid in hepatocyte, and the berberine also decrease the SIRT3 expression. In the presence of the pyruvate, the expression of SIRT3 is increased. However, berberine can decrease the expression of SIRT3. And in the presence of the berberine, the SIRT3 is not binding with the MPC1. In the pyruvate present, the SIRT3 is bind with the MPC1, but however, when they treat with berberine, they found that the binding between the SIRT3 and MPC1 is loose. And they continue to knock down the SIRT3 by siRNA, and they found that when they knock down the SIRT3 in the hepatocyte, the expression of the MPC1 acetylation is increased. However, in contrast, when the overexpression of SIRT3 by the transfection of vector into the hepatocyte, the expression of the acetylation of the MPC is decreased. This data suggests that the MPC1 acetylation is mediated by the SIRT3. We can see that with 3-type concentration of berberine can reduce the expression of MPC1 as suggested before, and they also confirmed in this figure. And they found that the berberine reduced the MPC1 expression via degradation, and they confirmed that when they treat with the MG132, this is a inhibitor. Protein degradation inhibitor. Because when the berberine treatment reduce the MPC1 expression, but when they treat with the MG132, the expression of the MPC1 is decreased. Furthermore, when the overexpression of SIRT3, the expression of MPC1 also further enhance expression. And in line with this finding, they also check the lactase level. Lactase level when the overexpression of SOT3 also be decreased. In contrast with the overexpression of SOT3, the MPC1 expression increase its expression. When they knock down the SOT3 by siRNA, the expression of MPC1 is decreased compared to the negative control. And the lactase level will be increased in contrast with the when the overexpression of SOT3. So this data suggests that berberine inhibits the MPC1 expression via degradation by inhibit the SOT3 expression. When they add the pyruvate into the hepatocyte with the viral type concentration, the expression of the phosphorylated PDH decrease, such as that it maintains the PDH activity. In contrast, the expression of PC is be decreased, be increased. And as I said before, when the pyruvate go into the mitochondria, it will be converted into the asynchronous controlled by PDH and go and convert into the osteolysis for the gluconeosynthesis controlled by PC. So they would like to check the whether when they treat the berberine, the pyruvate will convert into the asynchronous or convert into the osteolysis or not. And when they treat with pH pyruvate acid into the hepatocyte, the PDH, the phosphorylated PDH be increased, such as it reduce the PDH activity. In contrast, with the same concentration of pyruvate acid, is increase the PC expression, such as that the pyruvate acid had ability to induce the pyruvate go into the gluconeosynthesis rather than T-cell cycle. And more importantly, they treat with berberine into the hepatocyte combined with the pyruvate acid, and when we can see that the glucose production be decreased when they treat with the berberine. And they also treat with the UK 5099. This is the MPC inhibitor, and DCA is the PDH activator. And two types of chemical had ability to reduce the glucose production in the hepatocyte. In contrast, when they treat with the two type chemical, this is a SIRT3 activator, and this is CPI613. This is a PDH activator, PDH inhibitor. This type chemical had ability to reduce the beneficial effect of berberine in the reduce the glucose generation in the hepatocyte. And in the conclusion, I would like to summarize in the response to the pyruvate load, SOT3 improve the MPC1 stabilization and can induce the transport of pyruvate to the mitochondria. And the berberine can reduce the MPC1 expression by the by degradation this protein, and then reduce the hepatic gluconeosynthesis and reduce the glucose production in the hepatocyte and in the liver in the general. And this is my presentation. Thank you very much for your attention. Do you have any question? Because the figure is not clearly, and I don't know why, because it's also public in the famous article.
So statistically, we figure out of the function of berberine.
Sorry, I cannot hear you clearly.
So what's their main product over not using some material berberine, right?
Can you?
That's chemical.
Chemical?
Chemical name is berberine, right?
Oh yes, I think so. The chemical in berberine is we isolated from this plant in general. And in the paper, they also suggest this chemical has many biological activity in the such as the antioxidant or anti-inflammatory activity. Yes, this is a natural compound.
Anyway, this article is fun. There are some from natural material and their concentration, some chemical material can work in the cancer, right?
I did not search the concentration used as a drug. Yes, I think they just test some concentration when they inject this chemical into the mice. This is a 100 microgram per 1 kilogram mice, and they also treat some concentration in the cell. Let's see, 10 micromole, 1 micromole, and 0.1 micromole into the cell. So I think they have to they will be isolated this compound to the plant or by chemical from the company. I'm not sure.
Anyway, I think they're finding the material mechanism, right? How to working in the cell.
You
Is
mean
it right? My understand?
You asked me why they sure this chemical can work in the cell?
So there are purpose this article. They found some natural material and finding the mechanism on cell.
First, as show in the data in the paper, first one they check in the mice in vitro first, and then they found that this compound can reduce the glucose production in the mice. We check the blood glucose level. It's reduce the blood glucose level in the hepatic mice, and then they decide to treat to the cell and to the mimic the hepatic into the mind. They treat the hepatic acid, the fatty acid to the hepatocyte, and then they check whether when they treat the chemical into the hepatocyte, it can reduce the MPC oxygen consumption rate and some gene or lactase level in this cell. And they found that the MPC1 may be related to the function of the berberine in the cell, because MPC1 is required for the pyruvate go into the mitochondria. And they also check the berberine control the MPC expression at the mRNA level, at protein level, or at the post-translation level, and they found that it reduce the MPC1 expression by degradation. I mean, berberine reduce the MPC1 by induce MPC1 degradation. Yes.
I wonder their target digit start.
Target
Digit. Some cancer. Some digits.
Can you write? Can you write in the box chat box? Can you write it down in the box chat box? I don't know why the Zoom is suggest the remaining meeting time just 1 minute. So after 1 minute, I will stop the recording. Is it okay? Glucose production. You mean what is purpose? Because the I mean it's related to the fatty liver and the type 2 diabetes in general. Because glucose production is the one type indicator in the liver disease, in the fatty liver and the type 2 diabetes in general.
Okay, I wonder what's that.
Yeah.
Okay, thank you.
Thank you so much.